Hi, my name is James Widner, an undergraduate researcher at the Pain Stress Lab located at the University of Texas at Dallas, headed by Dr. Benedict Kolber. I'm super excited to be here today to talk to you guys about the project I've been working on this summer. So today we're going to be talking about pain, not just any kind of pain, but chronic pain specifically. Now, when looking at chronic pain, there's a few populations of nerve cells or neurons that are of interest, one of which are the calcitonin gene-related peptide receptor or calcurl neurons located in the central amygdala. Now, these cells are especially fascinating because they've actually been found to be lateralized in the central amygdala. This means that, in this case, the calcitonin gene-related peptide receptor cells in the right central amygdala have been found to enhance pain, while those in the left have actually been found in some studies to have the opposite effect. Now, when looking at a system like this, it's important to be able to isolate one of these subpopulations of cells to examine the effect that they have on the whole system. This can be done through eliminating one of these subpopulations through the use of a ribosome inactivating protein conjugated to the target of one of these receptors. In this case, you guessed it, CGRP, hence the CGRP receptor. So let's take a look at how we did this, and we'll begin with our hypothesis. We're hypothesizing that a direct injection of the CGRP saporin neurotoxin into the right or left central amygdala will lesion or eliminate the cells in that area and alter the responses that mice have in behavioral pain assays. So how did we do this? If you'll direct your attention to the left of the slide here, you can see a general outline of the project. First thing we need to do is establish a biomarker in the central amygdala. In this case, we're injecting a virus into that region of the brain that's going to show a fluorescent signal. After 13 days or so after the injection, the signal will be generated, and it's important for us to verify that we injected that virus into the correct location. This is done through a technique called immunohistochemistry, where sections of the brain are collected, mounted on a slide, and bound to antibodies. One of these antibodies will have a fluorescent probe attached to it that will allow us to visualize the signal there. Once I've gotten the targeting down for these surgeries, we'll begin with injecting the CGRP saporin. This will then, of course, go into the central amygdala and lesion those cells there, as we mentioned. Now, there's two ways this can be verified, one of which is using the same technique we did to verify targeting. The other actually includes looking at mRNA, which is much more specific than looking at the protein itself. This will be done through a technique called RNA Scope, which is visualized here on the right side of the figure. So let's take a look at what we found. In this figure here, you can see one of the first injections I've done with an M. cherry virus that appears red when fluoresced. As you can see here, the site of the injection is actually slightly below the central amygdala. After a lot of practice, though, and injecting this green fluorescent protein virus here, you can see that I've hit the spot right nail on the head at the central amygdala, and you can see all of the cell bodies expressed there, outlined in orange there. Once this is here, it's able to act as a biomarker for us after we inject the CGRP saporin, like I mentioned. After that saporin is injected, these cells should no longer fluoresce because they'll all be lesioned. Now, looking at some of the literature that uses toxins like saporin to lesion receptor cells, we've kind of determined an ideal dose, or at least a place where we can begin when making this dosing regimen. Dose of 0.048 micrograms per microliter injected over 90 seconds at a volume of 500 nanoliters. So what did we find from this and where are we going? We found that 900 nanoliters of the green fluorescent protein virus is sufficient for generating a biomarker in the central amygdala. Also, we've established kind of the beginning of the regimen for injecting the CGRP saporin. In the future, we're going to continue with the CGRP saporin injections into the central amygdala and obviously hope that they're lesioning the cells there to support our hypothesis. We're also going to optimize the RNA Scope protocol I mentioned earlier so we can more easily quantify the mRNA in those cells and see how well they're being lesioned. Finally, once we're successfully lesioning cells, we'll begin assessing the pain sensitivity of those mice that have had their cells lesioned in behavioral assays. Thank you guys very much for your time. I really appreciate it, and I was so happy to be able to work on this summer. Thank you.